The information contained in this video is intended to provide educational information and does not make claims or provide medical advice as it relates to any products sold by NViroMune or on this site. This video is for educational purposes only. These statements have not been evaluated by the Food and Drug Administration. This product is not intended to diagnose, treat, cure, or prevent diseases.
So it's my pleasure to welcome today Professor Ehud Gazit, who, amongst other things, is involved and is the founder and academic director of the Blavatnik Center for Drug Discovery at Tel Aviv University. And I think this position in particular, which relates to drug discovery, is an extremely relevant position with regards to what we are going to be speaking about today. We're speaking today about a formulation that Professor Gazit and his team of collaborators, Professor Danny Siegel and Professor Ron Bacharach, as well as their students and PhD graduates, have worked with to formulate a product that potentially has the ability to reduce the viral activity of COVID virus, but not only COVID virus, influenza virus, pneumonia virus, and all the common RNA viruses that are essentially a scourge to humanity. We live every day of our lives with viral possibilities that can infect us, that do infect us, that are seasonal, and that cause tremendous medical issues, death, especially in elderly people, and ultimately many days lost at work and school. And we've just really had nothing to deal with it. So the formulation of a natural product, and I emphasize the word natural because the product is made of so-called GRAS products, which means generally recognized as safe, is different to products that are pharmaceutical. And without me going on much further, I would like to ask Professor Gazit, who, if I may, is known by everybody as Udi, which I think his colleagues, I don't know what his kids call him, but everybody knows him as Udi.
Those are my kids. They call me the dad, but
it is an absolute privilege and an honor to have someone who is a giant in his field, has basically worked with the Minister of Health in Israel, and is considered a go-to person in so many areas. Professor Gazit, thanks for spending some time with us. I know how valuable it is. And why don't we just hear your concept of what you have formulated with your team? Tell me why you decided to go in that direction, and we'll just keep talking.
Okay, thank you very much, Norman. It's a pleasure to discuss this with you. As you rightly said, viral infection, especially of RNA viruses, and ways to treat this is truly unmet need. I've been involved in the last 2 decades and more in the area of drug development, and we've been working on all kinds of drug candidates, some of them already in clinical trials, for the treatment of various diseases. Yet, viral infection is truly a unique one. It's not so therefore, it is not surprising that we don't have good pharmaceutical treatment for viral infection, especially of RNA viruses. What was our starting point? We knew that zinc, which all of us are familiar with, some of us take it as supplements, is an excellent inhibitor of the proliferation of RNA viruses. Why? What is the basis for this mechanism? It's the ability of zinc to inhibit the process of RNA-dependent RNA replication. the way that the virus could replicate, and you get the formation of new viral particles, infective particles, and then the infections goes on and on. So zinc is well known to do it. So you could say we could take only zinc. Yet the problem, and the huge problem, was the fact that if you take supplement of zinc, which is generally regarded as safe, the intracellular level of zinc are not high enough. You need to combine it with ways to get the zinc into the cells. So
if I may interrupt you just so that it's very clear. If people go into their drugstore and buy zinc piconolate, but whatever zinc product they use, the truth is that what you're saying is that we just can't get the zinc, we can't take enough zinc to really block the viral activity that we should hopefully be trying to do.
Yes. So you can take zinc. I think that zinc is very safe. And actually, zinc is involved in so many enzymatic reactions in the cell. Yet, in order to inhibit the viral infection, you need higher concentrations. And this is where our invention, that is backed by strong scientific activity, comes into place, and we prove it experimentally, the idea that we have theoretically. We took polyphenols from plant source, also considered as safe. We based on a very solid body of research, which showed the ability of these polyphenols to insert the zinc into the cells. We combined the two together with additional ingredients that allow a good balancing between the minerals as well as the polyphenols. And we could prove actually two things. First of all, the ability indeed to get the zinc into the cells. We could show that you get the zinc into the cells, you inhibit the proliferation of the viruses, and we could test also the mechanism in which we demonstrated that indeed our ability to inhibit the proliferation of the viruses is because we inhibited the replication of the RNA genome of the viruses. So we had a theory that is based on very solid science. We test it experimentally, and we now know that indeed we have a unique composition, which allow the increase of the intracellular concentration of zinc, which inhibits the proliferation of the viruses, and that the mechanism in which it inhibits the proliferation is by affecting the replication of the virus. Why is it so important? First of all, it's a very generic mechanism. So it's independent of variants. It could be COVID-19 Delta, it could be COVID-19 Omicron, but it could be other respiratory winter viruses, influenza, a bunch of RNA viruses that are involved in most of the cases seasonal respiratory illness. So we have a mechanism that is generic, that is very effective, is based on natural compounds that are very safe, and it's insensitive to the formation of new variants, both of not only of COVID-19, but SARS-CoV-2 or the thing is the source for COVID-19, but also influenza viruses. As we know, every winter we have new variants of influenza, and our approach is independent of the new variants that are being formed. So this is why I like it so much.
So, I mean, that's really, I think, quite fantastic and needs to be emphasized that, and I don't know, when you were having the initial idea, did the pandemic of COVID stimulate this idea, or were you already thinking of creating an antiviral formulation before COVID?
So I've been working on viruses, on viral assembly for the last decade or so, in collaboration with Professor Baharach. We have several publications. We're mainly interested in the way that the viral particle are being assembled, and we have quite a few joint publications, highly regarded in the scientific community, published in top journals, in which we study the activity of viruses. And this is where our source of collaboration with Professor Baharach.
So when...
And on the other hand, we have a very long collaboration with Professor Siegel for the last 15 years or so, in which we're studying polyphenols and polyphenols in different type of contexts. And then bringing together our extensive and intimate, I would say even, knowledge of polyphenols, together with this concept of zinc-dependent inhibition of viral replication, bringing our knowledge of pharmaceutical ingredients, studies mechanism with a world expert in virology and a world expert in the biology of molecular biology, and our more biochemical background brought this really synergistic activity of all the different experts. So I think it's in that sense, it's also quite unique. We have here 3 experts in their field working together toward this goal. And then I think this was also the, I know everything about polyphenols, but I don't know how to grow the viruses in my lab. And this is where we collaborated with Eran or Professor Baharach.
You know, having spoken to the other professors, I think, quite honestly, this is a totally unique collaboration. As we all know, it's very difficult to get different professors, different departments, different interests together to collaborate in such a rapid fashion as you 3 professors have done, to create a product that really is almost a silver lining of the COVID pandemic. As tragic as the COVID pandemic has been, I have to believe it was a catalyst for you all to really put your heads together and say, look, this is an emergency, urgent crisis. We've got to get this done now. You'd been doing it, as you said, for a number of years, but it hadn't been formulated. Now, in a very short time, you've put this together.
Yes, I have to say, it's for me, as you mentioned, I served also on other national level positions regarding to science. This was also our duty. As soon as we realized the extent of the pandemics, graduate students in the lab who in parallel continue to work on other topics that are more in the core activity of the lab, pull together their effort together with us, the 3 professors, and invest much of their time, our time, in order to make it so quickly and really to be very focused on the ability to make an effective formulation to really understand, to prove its activity and to understand the mechanism of it.
So let's talk about the study. So first of all, we've already distinguished that this is not a pharmaceutical product, which I think is very important, because obviously, and you may want to comment, pharmaceutical products take much longer to develop, have much more potential for adverse events. This is a natural product that's readily available and can be formulated. So that's a huge distinction.
Yeah, this is each direction has its pros and cons. I'd say ability to use natural products in the case that it's active. You can be in a case when you have natural compounds that are not as active as new pharmaceutical ingredients. In the situation in which you have a new pharmaceutical ingredient, in the case when you have natural product that is as active as a new pharmaceutical ingredient, and when you are using your brains actually to do this combination. So it's not just taking a molecule from a natural sources, plant source or other, and take it, but the unique thing here is the combination, which is also makes it very complicated to get a regulation in terms of a simple pharmaceutical product. I think that this is where natural products have advantages. We are using products that are being used by humans for many years. The safety is well established, and if we see the safety and thinking about this clever combination, and you could see the efficacy in viral proliferation via the inhibition of viral replication, this is the best thing. Not taking the place for other things that you might need the development of pharmaceutical new pharmaceutical ingredients in which you don't have this option to get the natural products. But this is a unique case, which I think it reflects the two things: the fact that polyphenols are such outstanding ionophores. This is the scientific terms for compounds that could bring ions, metal ions, into the cells, may be part of a physiological activity, together with a combination with these ions, these metal ions that we include in the formula, included in the formula.
So what I'd like to talk about next, so we've obviously summarized that we've managed to create almost a super zinc formulation where by using the combinations of polyphenols, we've managed to get zinc to a concentration where it's much higher, where you couldn't normally absorb it from standard zinc. It enters the cells, it stops the sort of viral cell replication. Can you tell me a little bit about the in vitro experiments you've done with the cells from the lung, where you've actually looked at the ability of this formulation to actually do exactly what you've described, block viral entry and block viral replication? How did you do that? So
we're following the proliferation of the viruses. We could clearly see a reduction, a significant reduction in the number of viral particles that are being formed using standard assays in collaboration with our excellent virologist, Professor Bachara. So based on the clear scientific methods to quantify the number of viral particle, we determined that a statistically significant reduction in the number of active virus particles. And then we went on to determine the amount of genetic material, how much genetic material is being made. And we really could see using PCR, just the same, the quantitative PCR. Now, until 2 years ago, nobody knew what PCR, well, outside of the scientific field, what PCR is. But now, using PCR is a bit modified than the version that you do for the detection of virus, but still a very sensitive method in order to determine the amount of genetic material. We could see the remarkable the almost complete halting of the replication of the genetic materials. So we have an assay in which we determine the number of viral particles, and we could see its reduction. It was on the cellular level, so we don't have the entire mechanisms that you have in the full body to completely eradicate the viruses from the system. But at the level of the number of new genetic materials that is being formed, it's reduced almost to null. So this could be proven clearly in the cellular level.
Sorry, I just want to clarify for people who are listening and looking and watching that these were in the cells of the lung. So these were actual alveolar cells from lung tissue that was basically human lung tissue.
Yes, this is done in the environment in which the virus is infecting and replicating. And we could demonstrate in the specific tissue, in the relevant tissue, the ability to reduce to almost zero the ability of the virus to replicate. And means that we could stop the infection at the cellular level, and for individuals in which the replication is completely stopped, then the body is getting rid of the amount of the virus that is still there, and we could expect a situation in which
Let me ask you a question that I have no answer to, and it's once more, it's a speculative question. But if we get rid of the virus in the lung, do we think there's a time frame? Because we know that this virus can spread very, very rapidly to other areas in the body. So we're assuming that what it does in the lung, the formulation of the zinc, what it does in the lung is going to hopefully do the same thing to any other viral particles that may have moved on to other areas of the body. I think possibly up the olfactory nerve, even into the brain, is a very possible transport direction that this virus may take.
Yeah, I'm less of a virologist, but I can speak from the biochemistry point of view. I think as soon, and I'm extremely convinced by the results, as soon as you can stop the replication, there is no new material that allow further infection of new cells and the spreading as you suggested. As far as I see it, if you can do the extrapolation, of course, you have to take all the caution, but it's reasonable to speculate that if you inhibit this replication, as soon as you are infected, you can stop the spreading of the virus. So I think that if I extrapolate from the biochemistry to the virology, I think that it's critical to stop it as soon as possible, as soon as you know that you are infected.
Well, that brings us to a very important point, and I know that no one knows the answer yet. It'll come out in the future. The question is whether we should take this formulation, and I'm going to use the word NViroMune because that is now the official name for this product, NViroMune, whether it should be taken just when you get sick, when you get symptoms, or especially in the case of people who have some form of immunosuppression, other autoimmune diseases, other coexistent morbidities, should they be taking it prophylactically on a regular basis?
Yeah, this is a very significant question. I have to say I'm not a physician, but rather a biochemist. But as far as I could say, and you asked me before regarding the composition of the formula, the fact that these are such safe compounds that are used by humans for centuries, and it is well established, both the consumption of zinc as well as the consumption of polyphenols, plant-based polyphenols from various sources is only beneficial, and this is proven over and over. So if a person is in a situation that is, he or she are sensitive, and you want to be in preconditions in which you interfere with viral replication as soon as the body encountered the virus, it is reasonable to speculate that this would be done with such safe compounds.
Well, I mean, you look at the habitual way people have used vitamin C, vitamin D, calcium. I mean, on a regular basis. I mean, vitamin C in particular has a mythology regarding its benefits. Whether that's valid or not, it's questionable. But the point being, I think there's already an understanding that if people, and we know certain people, I know in my practice that people who have immunoglobulin deficiencies, autoimmune disorders, who are on medications that suppress their immune system, the biologics, for example, they are extremely predisposed to infections, both viral and bacterial. So from a viral perspective, this seems to me something that safe, easy to digest, and may well give them an extra barrier of protection from the world we live in.
Yes. No, polyphenols in general, without this, our unique combination with zinc, polyphenols in general are well established as the antiviral activity. This was proven not by us over and over again. I think that indeed, and as all of you know, they are part of a very healthy diet. The polyphenols are found in some of the most in food that is considered to be most healthy.
Can you give it for the people who are watching? Which would you say are the foods that have the most polyphenol content?
So I think that green tea is number one. EGCG is compound. Green leaves, many of the fruits, colored fruits and vegetables. So most of the things that are considered to be fresh fruits and vegetables have some levels of polyphenols. Many herbs have high polyphenol content, like curcumin, and as well as clove. Actually, clove has the highest amounts of polyphenols of all spices, and using the Persian and Indian cuisines. So polyphenols are all around us. There's a very good correlation between food items that are considered to be healthy and their content of polyphenols, almost one-to-one. It's
Let me ask you, one of your most, I think, well-recognized and famous work relates to amyloid.
Yes.
Do you believe that we are going to see any changes or increase in that particular scenario due to COVID? This is a crazy question, but I ask this because we have seen patients who've had so many mental issues, brain fog, that one wonders what is happening, and one wonders if this is possibly a prelude to many people down the road having some kind of presenile dementia or some organic brain symptoms, because some of these people are really, really mentally and neurologically very severely affected.
Well, this is still under huge scientific inquiry. The connection may be not direct but indirect. It is well known that amyloid disease, so these are diseases and neurodegenerative disorders that are related, usually related to old age, like Alzheimer's disease, Parkinson's disease, ALS, and others, could be affected by inflammation and pro-inflammatory activities. In general, having viral infections, putting the body into such stress could bring to imbalance of our system, and which could be part of a cascade that lead to neurodegeneration. It is less established with COVID. It is more established with other viral infections. Works not by us, but by other experts, show the correlation between herpes infection and Alzheimer's disease.
So
I don't think that it's direct, and I think that you could speculate that in a similar way, a viral infection, a persistent viral infection, which puts the inflammatory system into imbalance, could affect such conditions.
Well, what I was going to say is, when I grew up in terms of as a medical student, we always associated the correlation between Spanish flu, which occurred in 1918, with the development of Parkinson's disease in a huge number of people 40, 50 years later. So it sounds to me like a similar, I don't even want to think about it, but I mean, a similar scenario that potentially from a pathological point of view could in fact be speculated. And once
speculate, but I think there's more data need to be tested. But based on what people saw with herpes, a viral infection, I wouldn't be surprised, but it still need to be validated. Well, we don't know many things about COVID-19 still. But I would like to stress, as you mentioned in the beginning, it's we are looking not only on SARS-CoV-2 or COVID-19 infections, but generally on all our respiratory RNA viruses. Unfortunately, there are a bunch of them, highly highly dangerous. Some of them are much more dangerous than the virus that causes COVID-19, and most of them are related to seasonal flu, other respiratory winter illnesses. So it's a big bucket, and
very interesting to speculate. And I think our bottom line at this point is that if we can show that we're to some extent inhibiting the viral consequences that we're exposed to, then that's at least something that is worthwhile to start trying to do. And we obviously don't know all the answers, but we can see the value of something that's natural and something that makes scientific sense. So it is very, very exciting. And we're running out of time for me to really, say much more, but I wanted to give you this opportunity to, is there anything that I haven't asked you or that I've missed or that you would like to say?
I think that I said, but I will just wrap up. I think we have here quite a unique situation in which 3 experts in their field coming together, join forces in order to tackle a very significant unmet need for the society. We spent a lot of thinking and then a lot of experimental work in order to validate our hypothesis. It was fully validated in the cellular level, and we hope that we'll bring new approach and to the treatment of COVID-19, as well as many other RNA-related viral infections.
Well, I must say that for me, and I've been practicing for 40 years. This is really one of the most exciting, projects that I've involved myself with. I relate this in part to when we started coming up with the biological therapy for rheumatoid arthritis and autoimmune diseases, and then the world exploded. And I think potentially we've got a situation that's actually much more simple, much less expensive. And that's an important point, because drug costs have become so astronomical that being able to come up with a product that is relatively affordable, I think is extremely important, because I have too many patients who can't afford their medicines.
Yeah. And I have to say, I finish each and every of my scientific presentation with a quote from Leonardo da Vinci, which is, "Simplicity is the ultimate sophistication." And this is my personal motto for more than 2 decades in which I practice science. I truly believe that simplicity is the ultimate sophistication, said by one of the greatest inventor of humankind. And this is also a very, it's simple, but it is sophisticated.
And I think that's a great point to end. And I think you're absolutely correct. We make life too complicated in many cases. And so I think we have a simple product that's probably been in front of our eyes for many, many, many centuries, that the three of you have somehow or other put your brains together and created. And I'm extremely excited and optimistic about NViroMune making a significant dent in viral infections. And I just really, I'm counting the minutes till we can really start seeing this on the marketplace. So I thank you. For me, it was, as I said in the beginning, and I'll repeat it now, it's an exemplary honor and a privilege to be able to spend this time speaking to such a giant of his profession, and such a humble, pleasant, just enjoyable person to interact with. So I really appreciate it, and thank you.
My great pleasure. Thank you for all the kind words. And,
thank you very much. We look forward to further. I look forward to visiting you in your laboratory, hopefully in the
yes. You're very welcome.